So I believe last Thursday we went through and just looked at all of the individual reactions and tried to identify what things changed. So now what we'll do is piece those out and look at the individual steps within them. So what I'm calling kind of our on-ramp into the citric acid cycle, we're going to convert oxaloacetate into citrate using the acetyl-CoA. Acetyl-CoA came from, where did it come from?
The coenzyme A, but it's acetylized.
Like chop that chop that.
I believe it is chapter 15. We're looking at coming from our glycolysis pathway. Okay, glycolysis converted glucose down into pyruvate. We then took pyruvate dehydrogenase and converted it across into acetyl-CoA. So that's our source of acetyl-CoA. That's ultimately coming out of kind of our last sequence of our glycolysis. There we go. Okay. Then we've got the oxaloacetate, and we're going to react those two species using citrate synthase to make our citrate. And this was one of the things that we did talk about that was difficult to see, so let's address it again. How did this reaction work? So what we would need to be able to do is identify where our carbons showed up in our product. So we got red and blue carbons. Where are red carbons in our product? Or our blue carbons, I don't really care. Which one? Okay, so we've got 4 carbons capped by carboxyl groups. So one option that you might pick, which is what I happened to pick, was picked, or tried to pick right up the middle. 1, 2, 3, 4. If we go straight down that middle, that doesn't quite work, because then I've got these 2 carbons separated from each other, and the only source that I would have for those would be the blue carbons from our acetyl-CoA. And that really doesn't make sense to split those like that. So that method didn't work, which is fine.
You know your bottom 2 carbons on the oxaloacetate are your top 2 carbons.
Okay, I can accept that. So we're saying those 2 are those 2. Where's the next 2?
Middle carbon and one to the right. Kind of like horse and chest. The L shape.
Like a horse? Like how the knight moves, you mean?
The horse. You know, those
Yes, I see. I got you now with a horse, yes.
Either way.
Yeah.
That's just
Okay.
carbon.
That's our shape, right? How a knight moves, or the horse moves in chess. All right, so what does it move forward? 2 over 1?
No, I thought it was
1,
3
2.
over 2.
I think it's just 2, 1, isn't it? Either way, still pretty close to our overall shape for how a knight or a horse would move in chess. Okay, so those are our carbons from our oxaloacetate, which is then references where our carbons from the acetyl-CoA.
So,
It's then the bottom 2. I hear you, just a sec. Yes.
So why wouldn't it be the COO on the product in the middle of it? Why wouldn't that be the COO? Because the C in the oxaloacetate, the top number 1 or the top in the chain is connected by double bond to an oxygen.
So we're saying that one.
Yeah,
Why is that carbon? Which carbon in the product?
the center carbon,
That one.
yeah.
They are.
Okay.
They are one and the same. So if we look at this molecule, there's a lot of symmetry associated with it. If we draw a line right through the middle there, we've got symmetry.
Right.
So what we could have done is said that those 2 top, what we've labeled as our red carbons, could have been the bottom ones. Okay. You guys started saying with those being the red ones, so it doesn't matter. So I didn't care, and that's how I labeled them. If we tried to match the shape of our oxaloacetate from the beginning, we could just as easily have colored it the other way around.
I see that.
Yeah. Okay, and then this is our carbons from our acetyl-CoA.
Because I see it coming in from the bottom and pushing that up.
So everybody's going to see it slightly differently. This is one of the reasons why we addressed it, to see what's going on with that and see where those pieces are. So when we look at citrate, it's a nice relatively symmetrical molecule. So the reason why we're identifying those individual carbons comes from the next part on trying to put these two together. Okay? So as Diana was pointing out, that one carbon is the same in both cases. Okay? What charge is that one carbon in the starting material? So if we look at our starting material, we're saying over here, that carbon. What charge is that carbon most likely to be?
Partial positive.
Is it a triple bond?
It is not a triple bond, it's a double bond.
Partial
It's going to be a really strong partial positive. Why would it be a really strong partial positive? Because of the pi bond to our oxygen. So if that carbon's positive, what charge does our blue CH2 need to be to come in and react with it? Negative. So now what we've got to do is go back to our acetyl-CoA and say which of those two carbons is most likely to be negative. Okay, well, we got two choices. So 50-50, left or right?
Right.
It's gonna have to be right.
Okay, it's going to have to be the one on the right. Why is the one on the left not a good option?
Partial positive.
It's also partial positive due to our carbonyl.
You have sulfur that has the ability to donate electrons to it.
That carbon is, so it's not as positive, okay, but it's still going to be positive, okay, because of that carbonyl. Which then begs the question of how does that carbon on the right become negative? Because right now it has its full octet and it does not have a negative charge. So how does that carbon become negative? Okay, so let's see if we can draw that structure a little bit bigger. Sort of the resonance. Okay, that chart or that carbon right now is neutral. For it to become negative, what do I need to do? It needs electrons. Okay, so I can't bring electrons into the structure because then I exceed the octet. Okay, so what that carbon needs to do is take electrons from another atom. Okay, from an atom that it's bonded to. We've got 4 bonds to look at. 3 of them are hydrogen, 1 of them is carbon. Would it likely take electrons away from the other carbon? That's not a good option because that carbon is already positive. Okay, we get to push it further. It's actually sp2 hybridized, which means it's more electronegative than this carbon. Okay. All of that suggests that our terminal carbon needs to take electrons from a hydrogen. Okay, well, is that a valid maneuver? What's the result? We'd get our negative carbon and we'd end up with H+. Well, is H+ something that's reasonable to form
It will.
in the body that neutralizes acids and bases? Yeah, okay. H+ is not a bad thing to have coming on and off of a structure. As long as we don't get a ridiculous amount of it concentration-wise, the buffer system will neutralize that and we aren't stressed about it. So we can pull off H+ without an issue. Okay? That, as far as biochemistry is concerned, is relatively stable. But we now have an issue with that negative carbon. Okay? What have we learned from organic chemistry when it comes to negative carbons? Highly unstable. They are very, very rare to come across. That's why the most famous negatively charged carbon is named after the person that discovered it, Grignard reagents. Okay? So why am I allowed to form this structure? Came back to what Alyssa was suggesting.
If it's a Grignard reagent, water everywhere.
It's not a Grignard reagent.
Well, because of resonance, because you can push those electrons back out to the.
It is a resonance stabilized structure. Okay. We have an alkene and oxide, which is typically referred to as an 8. We have an enolate. Okay. That enolate resonance stabilization stabilizes that negatively charged species enough that it can exist in real solutions. Okay. That's what makes acetyl-CoA, that hydrogen, actually relatively acidic, even though it's bonded to carbon, which doesn't seem to make sense, but we have resonance stabilization of our conjugate base. That stabilization facilitates that hydrogen to be removed, and we now have a negatively charged carbon. Why is that important? What did we need that negatively charged carbon to do? To react with that positively charged carbon of our carbonyl. Right, but then that carbon has too many bonds, so what happens? We can break the pi bond, and we end up putting a negative charge on that oxygen. What would we need to stabilize a negatively charged oxygen? Say maybe an H plus that we'd kicked out earlier.
How can we get rid of the hydrogen though? Because doesn't it rarely happens, and we more facilitate that than just.
So, and this is where we run into kind of fun stuff with biochem. We might say that that reaction is not particularly favorable to remove that hydrogen. And in an organic solution, you get a very small amount of this process actually happening with that species. Okay? And that's because it is fairly unstable. Okay? We still can get it to work. Okay? But in organic solutions, it usually takes a little bit of time, and we're relying on the equilibrium balancing out and kind of slowly getting that species out and active. But we're not in a standard test tube for organic chemistry. We're in the enzyme. What is the enzyme doing? Lots of other stuff. Say a basic functional group that can come up and pull the hydrogen off. It can then transfer that hydrogen over to our other oxygen. So the enzyme is facilitating all of this overall chemistry by bringing in amino acids that will hook up and connect those, that will help polarize each of those bonds even further so that it reacts even faster. Okay? That process is due to the enzyme and the amino acids involved in it. And that's part of one of the things that the writing assignment was trying to get at, is look at your overall reactions on what things are changing. What did the enzyme do to supply that? Okay? What else is happening in there to help those reactions along? And in the papers that I looked at, that's what I was seeing, is people saw those reactions or saw serine was involved because serine is a reasonable nucleophile. Okay? Or an aspartate ion was involved because it has a strong negative charge, and that negative charge can attract a positive charge, okay, and help with ion pathways. Okay? So that's one of the aspects that's happening within your enzyme, is you're doing all of these kind of shifts around. And so that's why we talk about it at the beginning of the semester, and we're now shifting into kind of the later part where we're looking at the chemical reactions. Your enzyme is facilitating all of this by providing electron donors, by providing bases, by providing acids to facilitate that ultimate change in structure all the way across. Okay? So it's kind of a neat process. Okay? Within this, do we do any oxidation or reduction? No. Which means, do we need any oxidizing or reducing agents to facilitate this transfer? No. So we don't need any D+, we don't need any DH. We don't need to worry about it.
Carbon gains an electron though. So.
But other carbons lose electrons. Your net result is no change. Okay? So in that step, we've now converted our oxaloacetate into citrate. We can now kind of initiate our potential reaction, moving on further through our citric acid cycle. Okay, so in the next part, our enzyme is I forgot to look at this up, make sure. I think is aconitase, so that cis-aconitase may be questionable there. Okay, your enzyme does both of these steps. That single enzyme does both of these steps. Okay? Even though we see these kind of reaction arrows in between, it's because we form a nice intermediate that technically isn't an intermediate, it's a stable product. The stable product can then react further using that enzyme to take us all the way over to our D-isocitrate. And one of the issues with this mechanism, or one of the issues with this reaction, is noting a big, big change within this. Okay, what is that change? Oh, actually, it's even worse than that.
The hydride shift?
All right, we'll talk about that as well. Let's push it a little bit further. What's special about those red carbons?
sp3?
They are sp3. Because they're sp3, we have to consider something. What's that something we have to consider? Chirality. Citrate is achiral. Okay? If we look at that carbon in the middle, yeah, we have 4 things attached to it, but the 2 things on the side are identical to each other. there is no chiral atom in citrate. Through the process of doing this reaction, when we end up at isocitrate, we have 2 chiral atoms, okay, which means we have chirality associated with this, and you'll notice that what do they put out in front? D, in reference to the chirality of that product. Okay, we're not going to form the L isomer. We're only getting the D isomer out of this. Okay? So let's evaluate our overall steps in this reaction. What happened? So we look at our first step. What was the net result? What type of reaction? We have an elimination reaction. How do you know it's an elimination? Did the formation of a pi bond. Okay. How did that reaction occur? Well, what things changed? The OH went away. Okay. How does it go away?
By forming carbon.
Okay. It will take the electrons with it. Why would the oxygen take the electrons? It's electronegative. Okay. Oxygen is more electronegative than our carbon, so it should take the electrons. That does start to form a positive charge on our carbon. Okay. To form a pi bond, what else do I need? Sort of. I need electrons. Okay. At this point, all I have is a positive charge. To make that pi bond, I need another pair of electrons. Where does that other pair of electrons come from? A hydrogen. We go through and actually draw in our hydrogens. There's 2 hydrogens versus 1. We're taking the electrons away from the hydrogen to make that pi bond. There's our elimination reaction. Okay. Not technically a reduction or oxidation reaction. Okay. We've swapped one electronegative element for another. Okay. What happened in the next step?
Well, the OH.
Sort of. The OH does come back on, so and you're still looking at the overall pieces. I want the overall reaction. What type of reaction here?
You gain a hydrogen back and OH.
We gained H plus and OH, which means we did
Hydration.
in addition. We could call it a hydration. We hydrated the molecule by adding water back across the structure. Okay? This does introduce that chirality with this. It's also going to do something very important for us. Okay? Chemistry can now be very interesting about that position. Okay? And that's what we're going to look at in the next sequence of steps. So we've done an addition reaction where we added H plus and OH minus back across our reaction. There's our H plus. There's our OH. Okay? There's our addition. Okay? So the net result is really just the exchange of OH and H across our carbon atoms. We can't just pick it up and move it, though. We have to go through that elimination intermediate. Okay?
Does it make that alcohol group more accessible then, because you don't have sterics anymore?
What changed about the OH? Our OH is now a little bit further out in their structure. It's off of a secondary carbon as opposed to a tertiary. That secondary carbon is now more accessible to the solution. Okay, so what might we expect to happen in our next step? We're probably going to do something to that OH. Okay, so in our next step, we move from isocitrate to alpha-ketoglutarate. Okay, so good call on the OH. We did something to the OH. What did we do to the OH? What was that?
Oxidized.
Not just got rid of the H, we got rid of a second H. What did we do?
Took the electrons and picked off the hydrogen.
We didn't
Oxidized it.
oxid.
Is lost of electrons. Oxidation.
Sorry, I can't. Did I spell that right?
Yes.
That looks horrible.
That looks weird.
Yeah, maybe that's why it looks weird to me. I wanted to put a Z in there for some reason. We did an oxidation reaction at that point. Okay. What else did we do?
Oxidation because why? Because you lost the
So-
hydrogen and formed a pi bond. You added bonds to oxygen.
So that's the easiest way to look at it, is increasing the number of oxygen bonds. That doesn't always work. So if you can see a clear difference like that, then sometimes you can use it.
It's gaining electrons. Oxygen is gaining electrons.
It's all about the carbon. What happened to the carbon? The carbon technically lost electrons because
Therefore, the
oxygen will
carbon is
take more
oxidized, even
electrons.
though the oxygen produced.
Don't look at the oxygen.
Yeah.
What's your point?
Carboxylic acid also.
We also lost.
Carboxylic acid.
That's pretty close.
Decarboxylate.
We did a decarboxylation. Oh man, I don't know why I cannot spell.
OH.
Carboxylation. I think I just ran out of space. Oh no. Okay, so we did a decarboxylation. Okay. We've done this before. Okay, where we oxidized a position and we did a decarboxylation. Okay. This was done, I think we did this before, this was our pyruvate decarboxylase. Okay, so if we look at the enzyme involved in this, we're probably looking at a decarboxylase enzyme. Okay. This mechanism, to be discussed by classmates, sorry, I deleted some of the information, but not all of it. That was from the previous class. One of the previous classes actually looked at that mechanism. Okay. We aren't going to look at it in detail.
I was just looking at you've changed, is that still D-isocitrate though? Would you,
Yeah.
or is that your
I think-
isocitrate dehydrogenase?
So no, that's still your D-isocitrate. Did I do any rotations on it? No.
Okay, yeah, no,
You're
you're
right.
good. I
Yeah.
just wanted to, I didn't have to see it flow. Okay.
So now what we want to do is address how this reaction can occur. Okay, so let's look at the decarboxylase, because to me that one's a little bit easier to see. Ultimately, I know we don't have any charges out here, but that oxygen is negatively charged. So in the decarboxylase, one of the things we're spitting out happens to be, please reconnect. I think it reconnected. One of the things we're spitting out happens to be CO2. That CO2 happens to be up here. Okay? So these electrons must come down to form our 2 pi bonds of CO2. But then that carbon has too many electrons, right? Where do those electrons go?
Back to carbon.
We only have one choice of dumping them on the carbon, but now we have a negatively charged carbon. Is a negatively charged carbon stable? No. The only way we can generate that negatively charged carbon is if we have something to stabilize that negatively charged carbon. Okay, and when we look back at our conversion to citrate from our oxaloacetate, okay, we remember we used the acetyl-CoA, and we had a negatively charged carbon there. Why was that negatively charged carbon allowed?
It was stabilized with resonance.
It was stabilized with resonance with what functional group?
OH.
Not an OH.
No, it was carbonyl.
It was a carbonyl. There was a carbonyl at the beta position that facilitated that allowed that reaction to occur. Do we have a carbonyl yet?
Not yet.
No. So we said 2 big things had to happen. We had to do the oxidation reaction to form our carbonyl, and we had to do the decarboxylation. Which do you think happens first? Do the oxidation first. We can then convert this over into our carbonyl. Now when we go through and do the decarboxylation, it is resonance stabilized. All right. That resonance stabilization will facilitate that reaction. We can now do our official decarboxylation and our oxidation reaction. Okay? Kind of a neat process.
Is that where the intermediate of the oxalosuccinate or whatever is formed?
Yeah.
Right.
I don't know what they're showing in the book, quite possibly.
That's the intermediate with the carbonyl formed?
Yep.
Okay.
That is the intermediate they're talking about. Okay, so we have to do that oxidation first, so let's step back a little bit. How do we do that oxidation? What has to happen? clean that up. Careful, but yes. To form that pi bond, what changed?
Are you talking about making the C minus first?
Nope, we said the C minus cannot form first. We have to do the oxidation reaction. So we're looking at the alcohol turning to the carbonyl.
Yeah.
What needs to change?
One of the hydrogens pull away and the electrons shift.
Just say that again.
One of the hydrogens is pulled off and the electrons shift.
Okay, if we take, say, a base, we can pull that hydrogen off. That hydrogen should be very positive. Why? Because of the oxygen. So we can then take those electrons in and make a pi bond. But now what happens to that carbon?
It has too many bonds.
Too many bonds, so what do we have to do? What's that? The H goes away. What do you mean the H goes away? I agree with you.
You kick it out.
It kicks out as
H plus. H plus. Oh no, H minus.
H minus.
Because it takes its electrons.
It has to take the electrons with it. But H minus is not a stable species. What do we need to pick up that H minus?
An acid.
We need, it can't, then we're forming hydrogen gas. Then we'd all be combustible. That's a bit problematic.
Yeah, that's true.
You're right in that it needs to be positive.
So.
We need NAD plus. Okay. Because NAD plus can trap the hydride to form NADH. So one of the important side reactions happening is that we are forming NADH. Okay. That H minus is picked up by our NAD plus. Okay. We've now done our oxidation reaction. Once we've done the oxidation, we can go through and do the decarboxylation. We'll end up forming that resonance stabilized anion on the carbon. What does that anion need to permanently stabilize? A hydrogen. All it needs to do is pick up an acidic hydrogen that it can pick up from the solution. Understand which part?
They just did a poor job, I think, of showing that because it has the NAD plus coming in, but it doesn't show your H minus.
Where the
and
H
then
minus
now
is
it's
coming
showing a H plus after
Yeah.
you have NADH.
Where's that H? So this is something that bothers me about the NAD plus and NADH reaction. So what they're showing you is kind of the half reaction of this. Okay, so it's typically what they show is that overall reaction. Remember, this is being coupled with another reaction. What's the other reaction that it's getting coupled with? Okay, and actually I should balance that. We officially need the H minus here. So when we look at what happens to the NAD plus, it picks up the hydride, and this H plus just kind of shows up. Okay, so they're kind of showing the half reaction. Where's the H plus coming from? The H plus is coming from your oxidation reaction. It really bothers me as well. I don't know why they're doing it. I don't know what's happening with there.
Because
They're not showing where that H minus is coming from. They're not showing where the H plus is coming from. Okay, and it's because to go through and do that oxidation reaction, it's not just the loss of H minus. What else has to be removed? You have to remove that H plus. So when they're looking at that H plus just appearing, that's where it's coming from. It's coming off of your alcohol. Okay. So in this reaction, we need to consume NAD plus, produce NADH, and we can go through and shift this. Turns out the enzyme to do this is an allosteric enzyme, meaning its shape will change depending on what things are attached to it, which can either facilitate the reaction or prevent the reaction. There's two big inhibitors for this, ATP and NADH. Why might NADH inhibit this reaction?
Because you need to form NADH.
NADH is formed in the product. Okay? So if we're looking at all of these as being equilibrium reactions to a certain extent, the more NADH you put on, you're going to prevent the reaction from going forward because now you're blocking it, preventing it from coming out. Okay? So the NADH makes sense. What's going on with the ATP?
Yeah, that's a little,
Yeah, there's no phosphates shown in this reaction.
But
Okay.
the phosphate could react.
If you look at the entire Krebs cycle, you end up producing energy. You end up getting ATP, and GTP out of this. So the whole point of this is to produce energy. Well, if you already have energy, should you be doing the reaction? It's going to stop the reaction. So that's why ATP is inhibiting this reaction. We don't want to continue the reaction anymore because we already have a glut of energy. There's no point in continuing doing the reaction. That's why ATP is showing up. Okay?
What if it's like a low concentration of energy?
Now, if there's a low concentration of ATP, ATP is now no longer present. Let's take it to the extreme. It's no longer present. Is the enzyme inhibited anymore? No, which means the enzyme starts to function, and we can now start to produce ATP. Once we're producing ATP, now the ATP concentration can come up, and eventually it gets up high enough that it shuts down the enzyme again. Oh, yeah. There's the pieces that I thought were in there. So, through this reaction, we had that NAD plus involved, and we formed that enolate. Okay, we need that negatively charged carbon to be resonance stabilized, otherwise it can't form. Okay? Questions? Okay. Next step of the reaction, we're going alpha ketoglutarate to succinyl-CoA. What things changed? We do have this thiol group, and we also did a decarboxylation again. Whoops, I don't know why I circled it like that. But remember, if this decarboxylates, what charge would that carbon then pick up? Pick up electrons if we decarboxylated. If we go through and look at succinyl-CoA, the sulfur is a supply of electrons. Something really weird is going on here.
The sulfur attacks the carbon on the carbonyl.
The sulfur attacks our carbonyl. So we end up doing not just sulfur attacking the carbonyl, but we end up going through that whole thiamine cycle that we talked about before. Okay? You've got thiamine coming into this, you've got lipoic acid, FAD, NAD+, all of that big fun stuff. Okay? That was that big massive reaction we talked about before. I think that one was actually the pyruvate decarboxylase. Okay? So,
Can you go back one slide, please?
sure. Okay. So your thiamine goes through and attacks this carbonyl, okay, changing the chemistry about that position enough that we can then decarboxylate. The thiamine is then cleaved by reacting with the coenzyme A.
They're not showing any of the mechanism.
They aren't. They do technically show it earlier for a slightly different product.
Right.
Okay. So that, and I think I put the slide in and then immediately deleted it. That was dumb. We do have that slide earlier in one of our other presentations. It's just,
Mechanism.
did I? Actually, let's see, I may, yeah. It's this guy. No. So this right here, there's our carboxyl that we're trying to get rid of. There's the carbonyl next to it. This mechanism was for our pyruvate decarboxylase. What happened? The thiamine attacked our carbonyl. Right? It then went through a bunch of shift-based fun stuff. We brought in our lipoic acid to go through and do all of our conversions across to ultimately get us out to Acetyl-CoA. The Acetyl-CoA is the equivalent, oh man, now I gotta go back the other direction. Where'd it go? Well, nope. That is effectively what we have here. There's our CoA carbonyl, cover that up, there's our acetyl. Okay. So this enzyme has to be whoops, I meant to do the eraser. There's our Acetyl-CoA. So it's the same process, it just so happens that in this case, we're not looking at an Acetyl-CoA anymore.
It has another chain.
It has another R group coming off of it. Okay. It has to be a different enzyme. Why does it need to be a different enzyme?
So the different structure.
I would take it a step further than that. All of our enzymes grab a specific substrate. Does this substrate look different than pyruvate? Yeah, significantly different. So we're going to have a slightly different enzyme to facilitate that reaction. Okay. In step 5, we're going to take succinyl-CoA and turn it into succinate. What things changed in the structure?
Unless you're S-CoA enzyme.
The S-CoA was replaced with, you get the right idea. We have a carboxylic acid at the end, so let me redraw this a little bit. Maybe this will help.
And then a carbonyl as bonds.
What did the S-CoA become? Just became, the S-CoA is just the oxygen. Okay. Where did that oxygen come from? Nicely balanced.
The carbonyl, no.
Water.
When all else fails, it's always water.
Yeah.
Because
Waters are pretty awesome.
Yeah.
Yeah. Be all, end all. So water gets put onto this. We can then pick up an inorganic phosphate. We can then transfer that phosphate to GDP to form GTP. What is GDP versus GTP? It's a diphosphate versus triphosphate, effectively the same thing as ATP.
What does the G
Okay.
stand for?
Except the G stands for the other nucleotide. A stands for adenosine. G stands for guanine. Okay, I think it's guanosine.
Yeah, guan, yeah.
I think if we do the official name, I think it's guanosine.
Are there like allergy groups?
Yeah. Biochem just tends to remember the letters. It's a nucleotide, just the same kind of idea as ATP. So just like ATP can transfer phosphate groups around to change the energetics, GTP can do the same thing. Right? Why is ATP used primarily and not GTP? Good question. For anybody looking at evolution processes, we could look at why. Our half answer is saying that most of our enzymes are going to respond to ATP because the adenosine happens to fit in the proper enzyme pocket. But why do we have an enzyme pocket to fit adenosine and not guanosine? That gets back into the evolution of those individual
So
proteins.
would then different organisms have
You could
different pockets?
potentially look at different pockets, and yeah, it's a very slippery slope into the origins of life, okay, and deciding that. Okay. This is still mitochondrial. So if we take a look at our process, what we end up doing is converting this across. We end up generating an energy source through our Krebs cycle. We've got our GTP molecule. Okay. We're now at succinate as a structure. Succinate then goes through a big sequence of reactions through fumarate into malate. What changed? Succinate to fumarate. What happened? We formed a pi bond. How did we form a pi bond? Careful. It's not, I mean, yes, it is an elimination, but we want to be a little bit more careful with it because we can be more specific.
2 hydrogens.
And we got to be careful with that. It's not officially a dehydration either because dehydration is a loss of water, not H2. Okay. What we are removing is hydrogen. But if we got hydrogen gas, that's a bit problematic. What do we actually remove it as? What's that? We can pull off H+, I agree. So let's go ahead and do that on one side. We've got our H+, we now have as our intermediate an H-. Why would that H- be resonant, or sorry, as I give away the answer.
Right?
Why is that carbon now stable?
Because of resonance with the carbon.
Resonance with our carbonyl. Okay. So that gets rid of one H+, but then do I remove H+ out the other end? I can't remove H+ out the other end. Why not? Because then I'd have 2 negative charges. That doesn't work. So what am I removing out the other end? H-. How do I get H-?
Takes the electrons.
NAD+. Okay. Or I think actually in this case it's the FAD. Okay. So FAD will then convert to FADH2. Why would we look at FAD instead of NAD? NAD+ accepted what?
1.
H-. What does FAD accept? H plus and H minus. FAD is going to be our perfect transfer for this because it'll transfer both of those at once.
Because it's neutral to begin with, and then it's neutral
Yep.
to end with.
So if we draw our fun little coupling arrow into this, we start with FAD and we end with FADH2. One of those hydrogens has to come in as a negative. Okay? So we've done our elimination, which we want to be a little bit more specific, isn't just an elimination, it's actually an oxidation, because we've decreased the amount of hydrogens attached to our structure. That gets us to fumarate. Once we've got fumarate, what can happen? We then move to malate. What happens in that case? Careful. I do agree with both of those being valid. We did add an OH, and we did lose the pi bond. What else did you do? Added hydrogen as well. So we did a hydration reaction. Okay, yeah, and all these weird terms, hydrate versus hydrogenate. So what we've done is added water back across this. So we did an addition reaction in this case to get us over to the malate. Okay?
That's just that water was just like a byproduct, it wasn't part of the,
Where did the,
was it part of the initial?
you mean where did the H and the OH come from?
Yeah.
From solution.
Oh.
We didn't lose it anywhere else over here. Yeah, that's coming in as a secondary reagent to facilitate that. We'd have to add fumarate with water. So again,
So where does that pi bond
one of those things is very rarely ever shown. The pi bond goes to pick up the H plus.
Okay.
Okay, so let me draw a very, very, very crude mechanism. I fully admit that this is not a valid mechanism, so please do not give me a hard time on this. The pi bond comes out, picks up the hydrogen. The OH comes in. I guess I should show it going to the proper spots.
Yeah.
The OH comes into that place.
Okay.
Okay?
For our test next week, do you want us to like draw each mechanism or just?
What I expect you to do is to show me proper ideas on what's happening. So this is not a proper mechanism, but does it show that you understand what happened? Yes, that's perfectly valid. If you want to give me a full-on proper mechanism, I will fully accept that too. Okay? But all I want out of this is, can you show me where the electrons are going? If you show that those pi electrons get attacked by the oxygen, I'll have a problem with that. Why? What charge is the oxygen, most likely? Negative, because it's more electronegative than the hydrogens around it. Well, if you're showing the pi bond attacking the oxygen, you're saying electrons are attacking electrons. That's an invalid move.
Okay.
Okay? So if you can, and worst case, describe it.
Are you gonna have a step-by-step, or are you gonna show the reagent and then start reacting and then say, show me the product?
I'll give you reagents and products. I won't expect you to predict.
Oh, okay, good.
Yeah, yeah,
That's good.
yeah. If I'm looking for a mechanistic step, I will give you both.
And you want us
I
to
won't
know,
ask you to predict.
you want us to know the types of reactions, like oxidation is always
Yep.
when you end up with like more or less electrons than you were to start with? That kind of feels.
So,
Is that clear?
yeah, so what you want to do with that is evaluate your changes that occurred. So in this case, okay, we can't look at the number of oxygen bonds because the number of oxygen bonds didn't change. So one of the other things we could look at is for an oxidation reaction, it's increasing the number of oxygen bonds. It's also decreasing the number of hydrogen bonds. We've lost 2 hydrogens, okay, to go across that reaction. And if you're trying to decide on your reduction oxidation reactions, if you see a negatively charged atom that should not ever be negative, like hydrogen, that's a
Okay.
tip-off you're doing a redox reaction.
Okay. So increase in oxygen bonds and losing hydrogen atoms?
Mhm. And then we've got an addition in that last part. Okay, when we go from, I already erased the name, what did we say it was? Fumarate to malate. Okay? In our last step, we can take malate to oxaloacetate. Again, what things changed? Man. Spend your time looking at it fast. Okay. So this is a case where we could look at it and say what happened as we oxidized. If the hydrogens are drawn, it becomes a little bit more apparent as to what happened. We can take the electrons out to form the pi bond, but then we have to kick out H minus. Right, well, if we're kicking out an H minus, we've got to be doing some kind of oxidation reduction reaction, which means it needs to be coupled with a reduction oxidation mediator. That mediator in this case,
If it's only one, it's NAD.
NAD plus.
Now, where you're kicking out a positive and a negative, why wouldn't you use FAD?
It has to do with the enzyme itself on what it's facilitating in those transfers. The FAD is in the previous, let's see, so in this case, we have a very clear positive hydrogen. Let's see, I don't remember it was shown on this one. Yeah. In this case, do we have, whoa, no, come back here. Where'd it go? In this case, we don't have a clear positive hydrogen because we're looking at a hydrogen to a carbon. So yeah, in both cases, we end up transferring H plus and H minus, but your FAD is really facilitating transferring an H plus from a place that's not very acidic. Okay? Where your NAD plus will only pick up an H minus, it can't do anything about making a non-acidic hydrogen acidic. Okay? And it still has to be resonance stabilized. We can't just remove H plus from a non-acidic place. Okay? We still need that resonance stabilization to facilitate it.
So can you just say like if it's attached to, hydrogen's attached to a carbon, or if it's like non-polar, then it's
You're
FAD?
probably looking at FAD. Yeah. The other way to do this,
Because it's still two hydrogens that you're
Yes.
putting up, so there's no
but
clear.
one of them is a clear H plus, where one of them is not a clear H plus. Okay. The other way to do this, memorize.
reaction.
Okay. So memorize the important steps. Why do we have important steps? Well, wherever we're producing energy or we're using a redox mediator, those steps are of critical importance. The redox mediators end up getting used in the electron transport chain, okay, which is, granted, a mess of a chapter, but we'll be looking at that. And what you're doing is taking these electron sources, your NADH and your FADH2, and you're dumping those electron sources into the electron transport chain to facilitate effectively a battery. We then use that battery to produce ATP.
Okay, I have a question, but
Uh-huh.
it's maybe sort of a review.
No, it's fine.
So the hydrogen and the oxygen split, and they split heterolytically.
Yes,
That's
everything
why
we've
it's
done is
positive.
heterolytic.
The
Yeah.
homolytic, the hydrogen-carbon splits homolytically, and that's why it's negative.
No, if it's negative, it has to be a heterolytic
Yeah, 'cause there's
transfer.
both of them.
So,
Okay, so the hydrogen comes off of the oxygen grabs the electrons, leaving the H plus.
God, stop that. I thought I was drawing.
Yes.
Yes.
So
So if
the
we look at
hydrogen
this.
comes off of the carbon, but comes off as a hydride. Because it took
Well,
one electron with
so there's a reason why we aren't looking at the mechanism there.
it.
Because that mechanism could be done as a radical mechanism, in which case it is a homolytic cleavage. This is where one of the benefits of FAD is it can facilitate it as a radical mechanism, or it could do it as a heterolytic cleavage. Depending on the environment, depending on the mechanism in that specific location, it
So
may
now,
go radically, it may
If you go
not.
back one slide to the succinate and
Mhm.
fumarate, we have our two carbon. Right.
Yeah, I'm just trying to draw on the
So
hydrogens.
tell me again, why is, they're identical, why is one coming off as a hydride and one coming off as a hydrogen?
So that's going to come down to the enzyme and how it's facilitating each of those electron transfers. If FAD is involved in a radical mechanism, and so this could be our issue, do we know it's a radical mechanism? Do we know it's heterolytic? Okay. I personally try to avoid radicals like the plague because they're a mess. Okay. So when I look at this, I'm trying to look at the bulk changes at what happens. If I look at the bulk changes, I'm saying one of those comes off as H plus,
Doesn't really
and the
matter.
other one comes off as H minus.
Right.
Okay. That is one way to look at it. The other option is one, or they both come off as H radical. Okay. What does that do? Well, you're going to get the exact same result, except we now have to show half-headed arrows. One electron comes out there, one electron goes in. One electron goes out, one electron goes in. We still end up with the net result of the exact same things happening. Okay. So that's going to come down to what and how FAD is functioning. It can function as a radical initiator. It can also function as a heterolytic cleavage.
This makes more sense with my organic chem.
Okay, then stick with that. Okay?
So it's always hetero for these, all these, nothing splits homolytically except.
All
for
we've
the-
looked at is the heterolytic cleavage because the heterolytic cleavage tends to be easier to understand. FAD has the potential to do it as a radical cleavage. How is FAD facilitating this? Is it doing it as a radical cleavage? Is it doing it as a heterolytic cleavage? I don't know. Okay. So what we focused on first was what had to change. What mediator do we need to use that? Because we are now breaking these nonpolar bonds, NAD is not a good option. Okay, or NAD plus is not a good option. That's why we end up using FAD. So it's kind of our backhanded way of saying NAD doesn't do a good job, so let's use our other one, FAD. Okay. The other, even more backhanded option is what is used by the enzyme,
Yeah.
FAD. Okay. So we'll look at some of the mechanisms in detail, but we don't need to know them strictly that close. What we're trying to do is evaluate where those differences are, and when we see a difference in that electron path, that's going to change what mediator we're using. Does that make more sense?
Do we have any diradical mechanisms in our human body?
Probably.
Really? Wow.
Were they supposed to be there?
Um,
Probably not. Okay. It's going to come down to each of those pathways and specifically how they're doing it and what they're doing on it. One of the things that's going to make that more difficult to study, how reactive are radicals? Very, very reactive. So it's very difficult to detect those. So if we take the enzyme out and put it into a test tube, we're now missing all of those other species that could be facilitating the radical transition, but now we're going to see it as a anion or a charged transition because we've removed those radical mediators. Right? If we bring all of those mediators in, now it gets so vastly complex that it's virtually impossible for us to actually see anything within it. Come on. Okay, there we go. And we now end up back at oxaloacetate using, again, our NAD plus as our transfer. Okay. So we've got our big circle again with everything going through this whole process. There's a couple kind of important points that can be controlled. Okay. We've talked about some of these, and if you look at some of those pieces on what things control it, a lot of it has to do with ATP or ADP, okay? And a lot of that is because we're trying to either make energy or not make it anymore because we already have a surplus, right? So that's what's happening with your ATP and ADP. What happens with your NADH and NAD+? Why would this reaction be stimulated by NAD+? Well, what happened is we went from isocitrate to alpha-ketoglutarate. You guys think back? We needed an electron acceptor. So NAD plus is going to stimulate that because to do that transition, we need it to facilitate it. Okay. But you'll notice not all of them are inhibited or stimulated by those electron acceptors. We did succinate to fumarate, right, was our FAD transfer. Malate to oxaloacetate, okay, needed an electron mediator. Why are those not controlled? That's a trickier aspect, and inhibited versus not inhibited. Each of these cases where we see those little white lines through it are inhibited, or for that matter, stimulated by those other pieces. They have some regulation mechanism to them. The other ones do not have a regulation mechanism. So the question is, why do they not have a regulation mechanism if they're still using and producing the same stuff? Well, this one's right at the very beginning. In fact, we have two almost right at the beginning. In fact, these, oh, I see what you're saying. No, yeah, never mind. You're saying these aren't inhibited because they're more towards the end of the cycle. Okay, that's a possibility.
Is it because when you're going through the citric acid cycle, once you produce an ATP, basically due to feedback inhibition, I'm assuming that alpha-ketoglutarate could be used for other cycles and other processes of the body?
There's our primary issue. Some of these molecules are very important for other synthetic processes. Okay? If I need to use oxaloacetate to make something, okay, I don't want to consume it here. If I've got a surplus of energy but now I need to make something else, I don't want to immediately start this cycle back up because now I lose it. Okay? If I need alpha-ketoglutarate to make some particular amino acid or make something useful, I don't want to be constantly consuming it in this reaction. I want kind of a basal concentration of that. It needs to be involved in other metabolic pathways. That's our primary issue with these inhibition processes. Some of those molecules are very important to other pathways. If you think back to that big figure we showed where you could zoom in and see all of the metabolic pathways, and if you looked at all of those arrows, pretty much everything is interconnected. Okay? So it's not just this is producing energy and now that's the end of all those molecules. This is producing energy. Each of those new molecules has a very important pathway somewhere else. We need to get that molecule out so that it can be involved in that other pathway. Okay? Everything is interconnected. Okay? And that's what, this is back to our different things that inhibit or activate those. That's where we're getting into catabolism. Maybe we're generating these pieces and we don't want them anymore, so we start trying to destroy them, okay, get them out of the way because we have an excess of them. Okay? So there's no point in producing more energy because I already have too much energy, so I may try and turn those into waste. Okay? Or, now I got to jump way ahead in my figures. Or we can take those pieces out and use them to make something else. Okay? Anabolism. Okay? So for instance, isocitrate can be converted out to all of those individual pieces and we got glutamate. Well, glutamate was an amino acid. Okay? If we're not getting that amino acid from our diet, we need some way to make it. Okay? So we've got all of these different interconnected pathways. Your Krebs cycle is happening within the middle of your mitochondria. All of those little molecules could potentially come out and be involved in other things. Okay? So we want to be able to control where those pieces go. Okay? We go all the way back real quickly to our catabolism. So what this is just showing is how we can then take these things down to other molecules, okay? Or where we can supply the ingredients for this. Okay? So for instance, just because I happened to be pointing over there earlier, we got proline, arginine, glutamate, and histidine, I guess, can then be converted down into glutamate. Once we got glutamate, we can go to alpha-ketoglutarate. We now have a way of producing energy. Because now that I've got the alpha-ketoglutarate, that can go into the Krebs cycle and can produce energy. Why might this be helpful? What if you're starving, you have no food, you can't intake anything? Your body still needs to survive. Right? How does the cell survive if it has no source of energy? Well, it goes out to any other source. It can attack muscles. It can attack itself to generate enough energy to survive. Eventually, that's a self-defeating process, and you end up destroying the cell altogether because now it doesn't have anything to run. But it gets us temporarily through a rough state, if it is just a temporary state. Okay? So we've got to be aware of all of these different processes interconnecting through it. Okay? That's ultimately what's happening with our catabolic, is we're now self-sacrificing to generate energy so that we can maintain the health of the organism. And in anabolism, what we're doing is we now have tons of energy. Let's go through and produce all of those other side things that we need to function. Okay? So that's how we can go through and then look at all of those interconnecting pieces. Your textbook gives you a whole bunch of things on it, but really all I'd like to focus on is looking that these things can come out and be involved in other pathways. Okay? To generate proteins, to generate amino acids. Okay? We can then use those to survive and do other functions. Okay? So it's kind of a neat little process as far as looking at this whole kit and caboodle all together. Questions on that?
Would this be something that you would want us to label on a test?
Yeah, I'm going to go through and just delete all the words on that.
Not in the middle, but like, I mean, things on the
So,
outside. I don't know.
no, I wouldn't expect you to do that. Okay, what a question could be was malate, because we are looking at ultimately equilibriums between each of these.
Sure.
Could say malate is involved in the synthesis of, I can't even read what that says, pyrimidines or lysines. Okay, what might happen to Krebs cycle's formation of oxaloacetate if we needed to form those? Okay, well, if I need to make lysine, what's going to happen to oxaloacetate production? It should decrease because the malate's coming out to do that and not being involved in the oxaloacetate synthesis. Okay. Does it strictly decrease? Probably not, because there's other ways you can make oxaloacetate. Okay. But it's looking at kind of at the simple level, if we decrease this, what happens to this? It's going to decrease initially.
But then
That's
it'll
going to
pick up
cause
somewhere
a side reaction and say, okay, I need to produce that. Okay. But what we're looking at is that initial response.
Because it can come from pyruvate, is that?
It can come from pyruvate, yeah. So that is, where's that slide? That slide that your textbook goes through and does. So your oxaloacetate can come from a variety of different places. In plants, Acetyl-CoA can synthesize it. It can also synthesize your pyruvate. Okay. In animals, we can convert pyruvate into oxaloacetate if need be.
So what's the gatekeeper on that?
Gatekeeper on,
Well, you just don't want free oxaloacetate being produced if malate is drawing enough. I guess I'm just asking, where's the gatekeeper? Because if you suggest that in order to make the other lysine, that malate then is going to initially slow the production of oxaloacetate, then there must be a gatekeeper to suggest that it's not constant.
Wouldn't it just be a
How do I know that
feedback
oxalate isn't constantly being produced at the same rate, regardless of the lysine production?
so I think I see what you're saying. So what we would be doing is simplifying it ultimately to just these pathways. We wouldn't be looking at those secondary pathways because ultimately the concentration should stay for everything constant. Because if it doesn't stay constant, we're in an excited state and everything's going to be in flux.
So it's not technically accurate to say it decreases then?
Your initial state is going to decrease. So what will end up happening is another process will kick in to increase the concentration, to counteract that decrease.
Okay.
Okay. So if we look at just that process, malate going out and malate goes to oxaloacetate, and we say nothing else can happen, what's going to happen to the oxaloacetate concentration? It's going to have to drop. As it drops from the concentration from malate to oxaloacetate, another process has to kick in
I
to
get
replace
it.
that.
I just, this
Yeah.
is where I start looking at things, because we look at it, we break it down and we say, oh, well, this is the pattern that happens and it has to be sequential, but we really don't know if it's sequential.
No, we don't.
Because we're just hypothesizing for simplification purposes, because otherwise you could say, well, the fact that malate is producing this byproduct also stimulates this, so there's actually, and there's a homeostasis that is always present. We don't know if one is less
Well,
than or more.
We do know that the homeostasis can be destroyed, hence cell death.
Correct.
So we do make the assumption that we do fall out of homeostasis, that something, one piece fails. So we could look at some kind of degradation that says that destroys all possible ways of making oxaloacetate except from malate. Okay. That's going to be looking at some potentially cancer or some disease. Those enzymes are now destroyed. That is going to have an effect on this. Your oxaloacetate concentration will drop. And we can theoretically do that by mutating each of those enzymes to prevent oxaloacetate being consumed in other places. They are all linked, and in a perfect system, we wouldn't have to worry about that because it will maintain homeostasis all the way through. In a real world, it can't maintain homeostasis. It has to change. Which is why you stop running, too. I mean, you go start running. What do you need to run? You need ATP to power your muscles. Eventually, what happens? You stop running, okay, because you've lost that energy. So the ATP concentration is very clearly changing. It is coming out of homeostasis.
So I don't know if you can answer this or not, but like with
Probably
medications, there's the inactive salt form. Like, there's potassium citrate, there's enalapril, maleate, there's
Yes.
metoprolol, succinate. How does someone on those medications affect this system?
So what they're probably doing with those is trying to provide a source. So something about their metabolic process is either not producing enough, well, in that case, because they're ingesting it, it's not producing enough. So what we do is supplement that with a pill and hope that that then makes it through the digestive system into the bloodstream, which can then make it into your mitochondria and then facilitate those reactions to occur. So we can supply these ingredients from a variety of places. One of the things we talked about is doing it within the cell, okay? Destroying other things to make other pieces. But ultimately, our cells are not closed systems. They are open. Okay? Where are we getting glucose to start this whole process? From the foods we eat. It's not a closed system. We are bringing these things in, and supplying them. What happens to those things when they hit the cell or when they get into that metabolic process depends on what physical state they're in and where they ultimately need to go, and depending on the rest of that homeostasis system that the cell has tried to build. Okay? And as soon as we're whacked out of that homeostasis, that's when all of these things start to kick in and we see shifts and balances within each of those reactions.
What if someone's like gluten-free, so they can't take in glucose, so.
It's not, gluten-free is not glucose. Yeah, it's
It
a,
breaks down to glucose, that's a lot of carbohydrate.
there's other things in it. What is gluten? Gluten is the enzyme? It's not glucose. It's
something to do with it.
it's a different molecule. It's not glucose at all. Right, when you're gluten-free, I think it's an enzyme within part of the wheat plant that you're having an allergic response to.
So it's not like
But
an
it's not
essential
glucose. No,
part of this cycle.
no.
I have a question.
I briefly looked at it, but I
Very
don't
similar
think.
to what he was asking, like for instance, like a drug like metoprolol, for instance.
Okay.
I mean, like when you're using it for like high blood pressure,
Mm-hmm.
I mean, it's not just the metoprolol acting, so it's just also like the succinate part that is being like is being supplemented.
Yes.
It's not just to like stabilize the drug, the metoprolol.
Okay, now I'm confused. What did you say? Try that again.
Okay, so I mean, like the combination of like metoprolol succinate to
Okay.
treat high blood pressure, for instance. So the succinate part is not just to stabilize the metoprolol, so it's also as a supplement to treat
As a
the
secondary
disease.
drug. So what's happening is that both of those molecules are somehow involved in an integral pathway within metabolism that we're then trying to take advantage of to do the individual drug treatment. Okay. So that
So both
succinate
of them
could be
are
involved
working.
here, but yeah, well, it depends on, again, what they're trying to do with the drug. Okay, and then you can look at delivery pathways as well. What gets it into the bloodstream? Okay, so it could be something involved with that. Drugs aren't as simple as you just ingest it and you have it. Okay? For instance, you could look at, as a really simple example, salicylic acid. Salicylic acid is derived from the root of the willow tree, and that cures headaches, pain relief. Okay? What do we know salicylic acid as? Aspirin.
So that's acetylsalicylic acid.
But the active ingredient is not acetylsalicylic acid. The active ingredient is salicylic acid. Okay? What's happening when you ingest it? When you ingest it, the acid in your stomach cleaves acetylsalicylic acid into salicylic acid. Salicylic acid is then what makes it into the bloodstream. So one of the common side effects of salicylic acid was that it gave upset stomach because it drastically increased acid content within your stomach. So if we use acetylsalicylic acid, you've removed the acidic aspect to it, but you've removed it in such a way that when it hits an acidic environment like your stomach, you now reverse that reaction and you form a salicylic acid in smaller doses. Those smaller doses give you an increased likelihood of absorbing it across into your bloodstream, which then makes it to the active site or where it needs to actually act. So drugs are not as simple as I want to add malate. Because if I just add malate, consume malate, that doesn't mean it's going to make it into my bloodstream. So, and we'll definitely have to stop there.